So I'm going to talk about some of the work we're doing in biomarkers for Alzheimer's and related disorders. Next slide. These are my disclosures for consulting and research funding. Next slide. So our notions of Alzheimer's disease come from a framework called ATN, or Amyloid, Tau, and Neurodegeneration. And this is a scheme of how all of it gets put together. It started out being relatively simple, with amyloid and tau being the hallmarks of Alzheimer's disease. But as more and more studies of people with cognitive changes associated with aging have been done, we've realized, so go back to the previous one. We've realized that there are a number of other players that can interact with Alzheimer's disease. And so the Alzheimer's pathology consists of plaques, which are made of amyloid, tangles, which are made of tau, and you need to have both to meet a diagnosis of having Alzheimer's disease. Those can be the only findings in the brain, and in some people there can be other findings. There can be vascular pathology, strokes and other vascular changes, and then several other pathologies have been described. Alpha-synuclein can be aggregated in the brain. Alpha-synuclein is a protein that is part of Parkinson's disease, and sometimes alpha-synuclein changes accompany Alzheimer's. And there's another protein called TDP-43. I'm not going to give you its full long name. Wouldn't be helpful, other than to say TDP-43 is another protein that can also form clumps or aggregates in some people as they get older. And sometimes it can be a major determinant of memory changes. Sometimes it can kind of contribute. So a lot of things can be going on under the surface in the brain that help us try to figure out what could be the major and the minor contributions. And along with all of that, people can have changes in cognition that can be all the way from normal. You can have normal cognition and some degree of pathology through subtle cognitive decline, mild cognitive impairment, and dementia. Next slide, please. So what I've told you comes from studies of brain pathology. What we'd like to do is not wait until somebody passes to be able to look at their brain and say, aha, now we can figure out what's going on. We want to use things called biomarkers to try and map the ATN framework. And so biomarkers are tests that we can do by, for example, imaging the brain or drawing blood or obtaining cerebrospinal fluid, and then try and measure things that might be relevant to the pathology. And we've made enormous progress to try and map the ATN framework. Regarding the A part, which is amyloid, we can do an amyloid PET scan, or we can measure the protein that builds up in plaques called amyloid beta 42 in CSF or blood. Regarding tau, we can do a tau PET scan, or we can measure some forms of tau called phospho tau in CSF or blood, although those are a bit more indirect. Regarding neurodegeneration, there are lots of different things that are going on in the brain, and there are many, many markers we could choose to measure for neurodegeneration. One change, if one compares the gross appearance of the brain, this would be somebody on the right with normal cognition, on the left, someone with advanced Alzheimer's, and there clearly has been a lot of shrinkage or atrophy of the brain. So we can do an MRI to identify atrophy and specific pathways involved. We can do a particular kind of PET scan to identify how well the brain is using glucose, or we can use biochemical markers of neurodegeneration in CSF or blood, and the more common ones are things called tau or neurofilament light. Next, please. So this is what a PET scan for amyloid might look like, and a lot of research in studying amyloid through PET and other methods has resulted in findings that, firstly, we can detect amyloid very well through PET scans. There are degrees of buildup. And secondly, amyloid PET can become positive 10 to 15 years before anybody on a track to develop Alzheimer's may develop symptoms. One can read an amyloid scan clinically as positive or negative, or one can come up with a number associated with the PET scan. This is called SUVR. So these are some examples: somebody who has normal cognition, amyloid negative, normal cognition, but amyloid positive, MCI, mild cognitive impairment, but amyloid negative. This would suggest that something else is contributing to the MCI. MCI amyloid positive, very positive, and then Alzheimer's disease, dementia with the positive amyloid PET scan. Next, please. Tau PET shows pathways where tau, or regions of the brain where tau has built up, and usually tau PET shows a spread from areas in the medial temporal lobe of the brain, as indicated in A and B, to spread to wider areas within this area called the temporal lobe, and then eventually more widely throughout the brain. These are ways to map the tau pathology, and tau pathology correlates with the degree of cognitive impairment. This would be the mini mental state exam, a test we commonly use to try and diagnose dementia. And one can have some tau buildup with more or less normal cognitive performance, but in general, the more tau buildup, the worse somebody's cognition tends to be. Next slide, please. So amyloid and tau PET are very expensive, and they involve radiation. So we have other things we've been trying to develop. And we are now able to measure amyloid biomarkers in CSF and blood. So this is a cartoon showing the parent protein called APP, amyloid precursor protein, and the amyloid beta protein is cut out of APP. I've drawn a little square around the part of APP where A beta gets cut out, and the cutting involves generating a number of different peptides that differ in how long they are. They're called A beta 37, 38, and 40, and 42, for example. A beta 42 turns out to be a little bit longer, a little bit stickier, and it aggregates and builds up in the brain in Alzheimer's. Because it builds up in the brain, less of it is released into CSF and into the blood. And so we can take advantage of this by measuring either A beta 42, or we can measure a ratio of 42 against 40. Next slide, please. We can also measure biomarkers that tell us something about tau, the protein that builds up in tangles. It turns out that tau in CSF and also in the blood consists of all kinds of fragments. This is a cartoon showing what the full-length tau protein would look like. And these would be some of the pieces that have been identified in CSF through various studies. These letters are antibodies that are used to detect these different tau fragments. Nerve cells are always releasing tau to some extent, and in Alzheimer's disease, there is an increase in the release, and we can either show an increased level of total tau or some forms of tau with phosphate added that are called phospho tau, and these are increased early in Alzheimer's disease. Next slide, please. So this shows us how A beta 42 in the cerebrospinal fluid, or CSF, relates to changes on amyloid PET. So an amyloid PET scan shows the buildup of amyloid. So a higher number on an amyloid PET scan is a positive scan, or more amyloid. And so as you can see in this figure, the amyloid PET would be positive in this blue zone over here. CSF A beta 42, or the ratio of 42 over 40, is normally relatively high. However, if amyloid is building up in the brain, A beta 42 decreases. And if you look very carefully, you get a sense that, in fact, the CSF may change a little earlier than the PET scan, and that's been borne out by a number of other studies. But in general, there's a strong relationship between finding a lower CSF A beta 42 level and higher amyloid deposition in the brain. Next, please. So one of the more exciting developments in the last 2 or 3 years has been being able to try and detect some of these biomarkers in the blood. Regarding A beta 42, it's been very difficult. There are several different methods, the best of which has been developed by a company in St. Louis called C2N. And to get the best results, they have to measure A beta 42 and A beta 40, and they have to compare them as a ratio. And the top figure shows the difference between someone who would be amyloid positive by PET or amyloid negative by PET. So if you're amyloid positive, this ratio is lower, and there's quite a bit of overlap. So if one also includes somebody's age and also their ApoE genotype, ApoE is a risk factor for Alzheimer's disease, then the separation is quite a bit better. It's still not as good as CSF, but not bad at all. And this can be used to try and predict whether somebody is amyloid positive or not from the blood. Next, please. And click on again. We can also measure some forms of phospho tau in the blood. This is one of them called p-tau 217, and a number of studies have been done in this particular study. Firstly, a number of people who had blood samples drawn and then died and had their brains examined were analyzed, and this showed that if you had more tangles, you had a higher plasma level of p-tau 217 versus people who had minimal or no tangles. Click again, please. And this was a group of different kinds, people with different kinds of diagnoses. P-tau 217 is increased in MCI. Click again. It's increased in MCI or Alzheimer's, and in fact, some cognitively normal people who are amyloid positive, and it is not increased in people who are cognitively normal or who had other kinds of neurological disorders. So again, a marker that can give us some extra information. Next, please. So a lot of exciting things are happening in the area of biomarkers, and there's some exciting developments in measuring things in blood. We are still continuing to collect CSF, which we do by doing a lumbar puncture. This is a cartoon showing that we introduce a needle into the lower part of the back, below an area where the spinal cord has ended. We draw off some spinal fluid, and that's kind of the story. It's a routine outpatient procedure to do a lumbar puncture. And the reason that we are continuing to do so is that the CSF biomarkers are far more accurate than the blood tests, for one. Secondly, we can measure far more than amyloid and tau. We can measure all kinds of things that tell us about inflammation, vascular health, other proteins. We've mentioned alpha-synuclein, and lots of other kinds of things that we're studying. And we can then compare blood biomarkers with this sort of gold standard of CSF. So we're making a big push to encourage people who are new to the ADRC to agree to a lumbar puncture. And people who are in the ADRC who've had one before, if you haven't had a lumbar puncture in the last 3 years, we would like to obtain a new one to be able to update our information, because people's brains change is sort of the the short answer. But also we're trying to correlate everything we do, the cognitive testing, the imaging, and so forth, with the most up-to-date biomarker information. Next, please. So talked a lot about all of the interesting proteins, amyloid and tau and so forth. We also are interested in vascular changes in the brain. This slide shows MRIs from somebody with normal cognition called healthy, someone with Alzheimer's, where there are changes we can see on MRI that show brain atrophy or shrinkage, and we can look at an area called the hippocampus. There are also some other areas where there's brain shrinkage, but we can also look at vascular changes in the brain, and there are a number of things we look at. This is one of the hallmarks, which is called white matter changes, and you can see these cotton wool-like changes within it's called the white matter in kind of the middle of the brain, and we can find these and other changes on MRI, and we're quite interested in extending our studies of this particular change. Next slide, please. And the reason that we're excited about this is that we're now part of a national study called Diverse Vascular Contributions to Cognitive Impairment and Dementia, or Diverse VCID. And we're going to be looking at these white matter changes in diverse individuals. This will be people who are not who are representative of different populations in the US. So some Caucasians, Latinos, and African Americans, and even some Asian Americans we'll be looking at. And so we've started to enroll for the study. So I'll stop there, and again, we'll deal with questions at the end. Thanks very much.
Hello, everyone. I'm Jeff. It's really an honor to be here to share some of my project with you and to give you some context as to how these resources from the ADRC are being used here at the Salk Institute. So in my capacity at Salk, I'm a postdoc with my own research project, and I'm also currently acting as the stem cell core director here. So I'm also responsible for taking some of the ADRC biopsies and making them into resources that are available to researchers at UCSD and at Salk. I want to give you a little glimpse behind the curtain as to how that works. Next slide. So for those of you who are not familiar, this is our beautiful campus. Unfortunately, I'm in the basement most of the time, but I do get to enjoy this view for coffee breaks. Our stem cell core is actually in the building in the foreground on the right, also in the basement. If you go to the next slide, I'll show you what the lab looks like and some of the resources that we have. So currently we have 2,500 square feet. This is a tissue culture facility, or TC, and where we have sterile hoods for culturing iPS cells, so that's induced pluripotent stem cells. We can make pluripotent stem cells from patient biopsy samples and expand the biopsies themselves out for various research projects. And we can also do long-term cryo storage in these liquid nitrogen tanks to cryopreserve our cells and then make them available for distribution. So if you go to the next slide, I'll tell you a little bit about where my project comes from and how I'm using these cells. I think Dr. Glasgow did a really great job underlying what is Alzheimer's disease and how our knowledge of it has come to be. About 95% of all cases are sporadic. That's random, as in there's no clear genetic basis for why someone gets Alzheimer's or not. And as laid out in the previous presentation, the majority of our understanding really does come from the clinic, although there's been really important fundamental genetic models in mice, for example, where we can take some of the known causes, genetic bases for Alzheimer's disease, and try to get an understanding of how these genes and how these changes are contributing to risk factors. But as we know, one of the greatest risk factors is age itself. And so that's one of the goals here, is to understand how a neuron from an aged individual that's cognitively normal differs from an aged individual that has cognitive decline or even Alzheimer's disease. And these resources from the ADRC really make this possible. So next slide. So in our lab, one of the things we can do is take some dermal fibroblasts. So these are skin cells from ADRC patients. And they can represent the entire human lifespan from young to very old. And we can directly convert those skin cells into neurons. So neurons of the brain that are the cells implicated in Alzheimer's disease. And when we do that, we can retain some aspect of age that comes from the skin. Conversely, we could reprogram those skin cells into a pluripotent stem cell, and in doing so, wipe away the age component and rejuvenate them, make them young. And then take those young cells, convert those into neurons. And so with this paradigm, we have two options. We have aged neurons, or we have young neurons. And in both cases, we can learn important information. On the one hand, with the fibroblast-derived neurons, we can learn what are the contributions of age. And with the fully reprogrammed neurons, we can learn what are the genetic bases that are built into neurons outside of age, and how do those contribute to Alzheimer's disease. So next slide. This is the paradigm I'm using for the data I'll show you today. So we take our fibroblasts from either a healthy aged individual or an Alzheimer's individual. We convert those directly into neurons to look at what the age component is contributing to the disease. And then we can take those neurons and do various assays with them. So for my project, we do RNA sequencing to look at what genes are being expressed and if they're different between aged, normal, and AD, and also metabolomics to see what proteins are there, what metabolites are in the cells, and get an actual concentration for those markers. So next slide. These are some previous results from our lab demonstrating the principle that I've described to you. So on the left, these are heat maps that show ages of fibroblasts from 0 years old all the way up to 89 years old. And the thing I will point out here is we're looking at changes in gene expression. So some of these changes are downregulation, some of them are upregulation. And at the end of the day, there's a difference, and it's age dependent of gene expression. And those are fibroblast specific. And then in blue on the right-hand side, we have the age neuron specific changes. And when we look at the comparison in the bottom cartoon, you can see there are some genes, so we had 71 in this study, that were associated with age in the fibroblasts, the skin cells themselves, and 195 that were specifically changed in aging in the neurons. So we have some ideas now of what genes are involved in this process, and we have a model in which we can look at human aging in a dish with real human neurons. But what are the functional changes? So one of the obvious choices was to look at metabolism. As we all know, our metabolism changes throughout our lives. And one of the main drivers of metabolism is the mitochondria, responsible for making energy. It's the currency maker for energy in our cells. And I'll just summarize this figure to the right in saying that the mitochondria and energy production decreases in our age neurons, in our model at least. And this fits pretty well with what we see in the clinic. So next slide. So we dug a little bit deeper into, that was old versus young, and now we're asking, what about healthy aged cells versus Alzheimer's disease cells? What are some of the changes that occur? So we're looking at transcription of genes and asking if these genes cluster into certain functional groups, and that's what these phrases are on the left-hand side. And one thing that popped out to me personally was a lot of these nucleotide metabolic processes. For those of you who are not aware, nucleotides are the building blocks of DNA and RNA. And it was really interesting because neurons are very long-lived cells. And so I wondered, what are these changes? And maybe are these some kind of precursor that we could use to understand aging in AD before the pathology onsets? So if you can go to the next slide. Transcription is all well and good, but it's nice to have actual concrete data of what the cells are doing. And so what we did was we performed mass spectrometry to actually quantify levels of these building blocks within the neurons, either from healthy aged neurons or Alzheimer's disease. And what we found kind of surprised us. So on the left graph, we found that on average, the amount of DNA building blocks available was significantly lower in Alzheimer's disease neurons compared to their age-matched controls. This has suggested that maybe there's some deficits in their ability to respond to DNA damage and stressors like that. So that's kind of where this research is going. And even more interesting, the graph on the right, we found that if you take the relative proportion of DNA building blocks versus RNA building blocks, so that would be long-term storage of information in the form of DNA versus short-term information transfer in the form of RNA, so gene expression, in other words, there's actually a much larger difference in this ratio, where we see about 100% more RNA molecules in healthy controls relative to DNA, but even more than that, double that in our Alzheimer's disease lines. So this is a little bit of a mystery at the moment, and we're looking to follow this up. As I mentioned, we have some DNA damage experiments ongoing, and just various metabolic pathways involved in these processes to see if we can identify one or two specific molecules that perhaps are druggable targets to start probing this pathway. Next slide. So that was all in a dish. And as Doug mentioned, we have all of the CSF available. And so I was wondering if there's any changes in nucleotide homeostasis in actual patient CSF samples. So we did a very similar experiment. We took the cerebral spinal fluid and performed mass spectrometry on it to quantify nucleotides. And we did find some significant differences in actual patient samples. So here is a nucleotide called uridine. And interestingly, we found it significantly reduced. Both males and females had some reduction, although it was only significant in the males. If you take the average overall, we still found a significant difference in uridine in the CSF of Alzheimer's patients compared to cognitively normal individuals. So with that, I will close out here. I got a little summary slide next. And I hope that I showed you that the resources available from the Adrc can be used to create human aged models of neurons in a dish. And we can use these to parse out differences in aging versus Alzheimer's disease to hopefully find pre-pathology markers, or just even understand the aging process in the brain and how it goes awry in Alzheimer's disease a little bit better. Some of our findings from our dishes have actually translated into CSF data. So the next step would be to ask if this happens, if it's just a bias in our small sample size, or if this is true in more patients. And then obviously CSF is a great tool, but blood is better. So we'd be interested to see if this is also a phenomenon that happens in circulating blood. And then, as I mentioned, obviously, future goals are to determine the cause and consequence of this. This is really just a phenomenon, an observation at this point. So to make it useful, it either has to be some kind of early diagnostic or a pathway that we can find a druggable target to restore function. And with that, I'd just like to thank you.
Good morning, everyone. I'm Svenka Slatter. I'm an assistant professor in the Department of Psychiatry here at UC San Diego. And I'm going to be talking about a little bit about the studies that we've been doing at the ADRC with Hispanic and Latino samples. That's basically what I have been focusing on. And more specifically, we've been looking at subjective cognitive decline and how it relates to objective cognition in older Hispanics and Latinos. So I'm very, very honored to be here to be able to talk about that. So next slide. So what is objective cognitive decline? It is an individual's perception of changes in their own cognitive function compared to a previous state and compared to other people of their age. And these are complaints that are self-experienced, and they may not really be captured by formal neuropsychological testing. So it is really a self-perception that your cognition or your memory function has changed over time. Next slide, please. So why should we study subjective cognitive decline? Because it may, in some cases, serve as an early risk marker of possible future cognitive decline. Next, please. So a group was convened. Oh, that figure does not look very good here. I don't think it's a little blurry. So let me just try to explain. A group was convened to try to formalize the definition of what subjective cognitive decline means. And the framework basically says that in this stage on the left side of the graph of what we call preclinical Alzheimer's disease, when people may have the disease in their brains, but there aren't any symptoms manifested yet. That is the time frame where this subjective cognitive decline may occur. And for some individuals, it may actually lead to progression to MCI or dementia, but not for everyone. So we really need to study for what kinds of people, under what circumstances, SCD could actually mean that they could progress to dementia, to see if it may serve as an early marker of or a red flag for clinicians. Next slide, please. So the key points in terms of defining this term of SCD has been that there is evidence that SCD, subjective cognitive decline, which I'll refer to as SCD, could potentially be a preclinical marker of Alzheimer's disease because it increases the risk for MCI as well, mild cognitive impairment. There is biomarker evidence of Alzheimer's disease in people who report SCD, and the people who report SCD plus have biomarker evidence of Alzheimer's disease have a higher risk of actually developing Alzheimer's disease. Our current knowledge is insufficient to comprehensively define the specific features of SCD in preclinical AD, so there are still many limitations. And SCD by itself may never be sufficient to diagnose preclinical AD, so that's really important to keep in mind. It's just something that we could potentially use along with other markers and other information, but not something that we would use by itself. And especially because there may be several other causes of why people report subjective cognitive decline, such as just being worried about one's cognition in normal aging. Other psychiatric conditions and neurologic disorders, may also manifest, people may feel like their cognition has changed and present with SCD. So things like depression and anxiety could also cause SCD. Medication and substance abuse and other cultural differences could potentially result in people reporting SCD that may not really progress to any neurodegenerative condition. Next slide, please. The problem is that this type of research on SCD as a potential early marker of Alzheimer's disease has not really been done with people of Hispanic or Latino descent in the United States. So we didn't really know much about how this concept of SCD actually applies to people from Latino background. So what we wanted to do was to study and determine if SCD relates to objective cognition in Hispanics, if it predicts cognitive changes over time, and if it relates to biomarkers of preclinical Alzheimer's disease in Hispanics. Next, please. So some studies that we have conducted have shown that there is an association between objective cognition and SCD in a large sample of Latino older adults. And that the graphs here basically just show that this ECOG that is on the horizontal axis is a questionnaire that people fill out about changes in their cognition as it compares to 10 years ago. And we can see that there are these negative associations, so that the greater people report SCD, the lower their cognitive scores tend to be. And this is, it was true for a global cognitive performance measure, an executive function performance, and a memory performance measure. Next, please. When we used that same sample and we separated them into individuals who are worried about their subjective cognitive decline versus those who are not worried about it. And the reason why we do that is because the SCD work group that define subjective cognitive decline thinks that, you know, when people worry about these changes, there may be more of a potential risk. And so that's why we separated them. We see that these associations with SCD reporting and cognition remain in people who are worried and those who are not worried. They are a little bit stronger for the individuals that are worried, but it's still significant for both groups. So basically what we are seeing is that these reports of subjective cognitive decline in middle-aged and older individuals who live in the United States but are Latino in background, they are associated. So this SCD is actually associated with objective cognition in this group. Next, please. And then using data from the ADRC from individuals from Hispanic background, we actually compared how Hispanics versus non-Hispanics had this association between cognition and SCD reporting. And what we saw is that for individuals who are from Latino or Hispanic background, which is the blue line, we see actually a negative association, meaning that higher reporting of these subjective cognitive decline symptoms were actually associated with worse performance on a measure of global cognition. And that was not the case for the non-Hispanic individuals. So that was an interesting finding. And so we're trying to learn more about why we see this difference in, why ethnicity is changing the association between actual objective cognition and subjective cognition. And that's what we're actually studying now using data from the ADRC. So next, please. We also wanted to know if subjective cognitive decline may be correlated with brain markers of Alzheimer's disease. Next, please. And we have very little data, so we're still working on gathering more data on that. But as you can see, we do see that there perhaps may be some association of subjective cognitive decline reporting with the volume of the hippocampus, which is an important area for memory formation, as well as other areas of the medial temporal lobe that are important for memory and that are affected in Alzheimer's disease. So basically, higher reporting of these subjective decline symptoms seem to be associated with lower amount of volume in these areas for Hispanics. Next, please. And so now what we're doing is we're collecting longitudinal data at the ADRC here in San Diego, as well as in the ADRC in Florida. And so we're going to combine data from the two centers to have a larger sample of Hispanics and Latinos. We're going to collect information on 100 individuals without any objective cognitive performance issues, and 100 with MCI, and as well as their informants or their study partners, because we want to learn about how their study partners' perceptions of these changes also relate to actual observed changes over time in cognition. We'll see if SED predicts change in cognition over 3 years among older Hispanics. We will investigate if it is associated with blood-based biomarkers of preclinical Alzheimer's disease, and also see whether culturally relevant variables such as acculturation, health literacy, country of origin, ethnic background, and language of testing can influence how people report these symptoms of subjective cognitive decline. And maybe that perhaps helps us explain those differences that we saw between non-Hispanic white individuals and Hispanic individuals. And I believe that is it for me. So thank you, everyone. And thank you most of all to the participants who make all of this possible. Thank you.
Good morning, everyone. Welcome to this next segment of the program, which is going to focus on treatment. I'm Howard Feldman, and it's my pleasure to give you a 10-minute tour of what's new and promising in the treatment of Alzheimer's disease, as well as in its prevention. Next slide, please. Here are my disclosures. I wanted to just mention that people may have seen in the news that we received a very generous donation from the Epstein family to accelerate therapeutic progress in Alzheimer's disease between two universities, USC and UCSD, and it goes by the acronym of the Epstein Family Alzheimer Research Collaboration. So by the next time I speak to you, I'm hoping to have some results to begin to share with you of the great work that's going to be done through this collaboration. Next slide, please. So there are two things that we're going to cover this morning. We're going to cover pharmacologic treatments for Alzheimer's disease, and in particular, Dr. Glasgow has done a lovely job stage setting, talking about amyloid scans and looking now at how are we doing at reducing amyloid levels in the brain and what is the clinical impact of doing this. There is a treatment that has been approved by the Food and Drug Administration called aducanumab or Aduhelm that is approved to remove amyloid from the brain. I also want to make you aware of the next generational treatments. These are the ones that we occupy ourselves at the Alzheimer's Disease Cooperative with every day, including Saturday and Sunday. And I'd like to make you aware of clinical trial opportunities within our UCSD environment through the ADRC. And if you're interested in learning more about any of the research studies that I'm showing you this morning or any of the trials, I give you the phone number and the contact email. Please avail yourselves of this information freely. On the prevention side of dementia, we're very excited to be launching a program called Healthy Actions and Lifestyles to Avoid Dementia. You see the Spanish translation. It goes by the acronym of HALT-AD. And Dr. Zlatar, who just spoke to you, is a driving force in that program as well. And I'm going to mention a lifestyle clinical trial opportunity in collaboration with Dr. Ornish. Next slide, please. So imagine for a moment that we do clinical trials, and imagine for a further moment that they run in stages. So there's a phase 1, a phase 2, a phase 3, and then there's an FDA review at the end of all of this. We cover trials at all levels within this spectrum. So presently, phase 1 are the very first times that we're exposing people to a new treatment. We will be starting a program called Modify AD with a gamma secretase modulator that will be running later in 2022. Imagine how great this is. We discovered this compound at UCSD through the efforts of Dr. Stephen Wagner and colleagues, and we will be bringing it forward with the idea that it looks very promising as an Alzheimer's therapeutic, and maybe one day even a prevention treatment. We're also looking at Posifen, and we're just completing a clinical trial of a very intensive phase 1 program. For phase 2 at our center, you may participate in treatments that are designed to lower the spread and aggregation of the tau protein that Dr. Glasgow showed you. We're increasingly looking for treatments that will hit multiple targets. So treatments will not only hit amyloid, but they may hit tau, they may hit inflammation, all at the same time. You heard Dr. Jones mention mitochondria, energetics, and metabolism. And we have a program that's going to start later this year looking at high-intensity dosing of thiamine to try and address thiamine deficient cofactor functions in the brain. And we've completed a study in this last year with troriluzole, some of you may have participated in it, that addresses impairments in glutamate neurotransmission. Moving into later stages, we get to phase 3, which are the stage just before treatments get evaluated by the regulatory authorities. There's some very promising amyloid lowering treatments on the horizon: lecanemab, gantenerumab, solanezumab, and ALZ-801. There's some multi-targeted amyloid treatments, donanemab. I'm going to show you a slide of that in a moment. And not to forget persons that have quite advanced symptoms, including disruptive agitation. We've been doing several trials, one with prazosin to block alpha receptors and one with a different kind of NMDA and sigma receptor antagonists to treat disruptive agitation. Eventually, at the very long line of research's conclusion, things go to the FDA, and we now have aducanumab that was approved in June of 2021. Let's move to the next slide. And I'm going to focus because I'm sure many people in the audience have questions about aducanumab, otherwise known by the trade name Aduhelm. It received approval in June of 2021. It has been shrouded with controversy and uncertainty. And if I'm going to boil it down for you, as I've tried to do in this slide, there are two parts of this. The first is, what do we know about the treatment? And the second is, what don't we know about the treatment? And hence, why is there uncertainty and controversy? What we do know is that this treatment, which is an antibody that lowers amyloid, has been tested in those in early stages of Alzheimer's disease. So that's what we do know about, is in early stages of the disease. We do know that it clears amyloid from the brain. So it achieves 31 to 48% amyloid negative from amyloid positive. We do know that higher doses are better than lower doses. We also know that, disappointingly, removing amyloid is not associated consistently with clinical benefit. There are two large clinical trials, one of which was resoundingly positive, one of which was resoundingly negative, creating a lot of uncertainty and leaving us with the belief that the clinical effects are probably modest on the beneficial side, if they can eventually be shown to be consistent. We also know that there are safety issues related to the use of this antibody. There are these things called amyloid-related imaging abnormalities, and I show you some example here. Here are two MRIs, and if you look carefully, you see this white high-intensity signal. This is brain swelling that can occur in a good number of persons that get treated with high dose of this. Estimates have been in clinical trials between 35 and 36%. Those with the E4 genotype are particularly at risk of this complication. Interestingly, 70% of the time, it doesn't cause any symptoms for the person. 20 to 30% of the time it does. It can cause confusion, headache, problems with walking and dizziness, with serious complications, meaning that people need medical attention, sometimes in the hospital, in 1 to 2%. So that's what we know. What we don't know is, what are the long-term clinical benefits? Are there any? Will this stop the disease course over a longer period of time, or will it change the course? How safe will this be outside of a clinical trial? You can imagine that being in a clinical trial attracts a lot of attention. People get seen regularly. There's a trial coordinator who receives calls anytime something happens. People are very closely monitored. In usual care settings, those same standards are not part of what we normally encounter. We really don't understand why the clinical effects are inconsistent, and also why the effect sizes are not larger than we would like them to be. There's an uncertainty over whether anyone's going to pay for this treatment, given some of these inconsistent results. When this first came out, you may have learned that this was $58,000 a year. We imagined that actually it would be $100,000 to administer it to a given person. We now have CMS that's evaluating the cost effectiveness of this treatment, and the preliminary indication is that they're only going to pay for it when they're part of clinical trials. So while the FDA has approved it, out of pocket makes this almost prohibitive to afford for most families and people. We also don't know, Dr. Glasgow's presented a lot about the biomarkers. There's this encouraging finding that you can lower phospho tau by 21 to 25% with this amyloid treatment, but we don't yet know exactly what that translates to. Move on to the next slide, please. So just briefly, part of the importance, I think, of aducanumab is that it's going to be followed by better treatments behind it. And here's an example of one called donanemab. It's a different kind of monoclonal antibody that works on a pyroglue amyloid target and also removes amyloid. And you can see here in this middle panel, by 24 weeks, you're getting quite a drop in amyloid, and you're achieving quite a high rate of amyloid negativity. So moving amyloid positive to amyloid negative is more efficient, seems to be more efficient, although this is only in early stages. We seem to see this relationship of a good effect on amyloid and on plasma tau. And as Dr. Glasgow showed you some tau deposition on PET scans, we begin to see some good effects on tau deposition with some lowering of tau levels inside the brain. This needs confirmation. This was a phase 2 study. It's now working its way through phase 3 studies, but there's a lot of interest in this. Next slide, please. So we mentioned a dual mechanism of action. So imagine for a moment that you could intervene before all the toxic forms of amyloid beta are formed. And we are currently testing through the ADCS a medicine called varoglutamstat. Imagine for a moment it reduces the toxicity of the amyloid forms that accumulate in the brain, as well as reducing some of the inflammation that occurs in the brain when we use this treatment in early Alzheimer's disease. So we're encouraged that this has dual targets. The site at UCSD through the Adrc is enrolling for this study. And if you're interested, please contact the numbers that I've given you. Next slide, please. Okay, we will be reading out in the ADCS a set of 4 clinical trials. And imagine for a moment, we've been working on these trials since 2013. So this really is an incredible year of accomplishment where we will have the results of an adult Down syndrome trial where we've used an amyloid vaccine. We have some encouraging results, and we have a paper in press with JAMA Neurology. I've mentioned the Posifin study, which we'll be reading out. The disruptive agitation study with prazosin that I mentioned will be reading out. And we're also reading out an aerobic training versus stretching program for persons treated for 12 to 18 months having mild cognitive impairment. So some of you may have participated in these studies. We look forward to sharing those results within this calendar year. Next slide, please. Okay, in the remaining last minute or two, I'd like to fill you in on what we're doing in the area of prevention. So we'll move on to the next slide, please. So why are we focused so much on prevention? Just look at this insert, which has really captured our imagination. 40% of dementia risk can be attributed to lifestyle factors. They run across the lifestyle continuum, or they run across the lifespan, from education, hearing loss, traumatic brain injury, high blood pressure, alcohol use, smoking, depression, social isolation, even air pollution has found its way onto the map. 40% of the attributable risk through these risk factors. And just imagine if we could reduce the prevalence by improving brain health, reducing risk factors, and delaying cognitive impairment. Next slide, please. So how are we going to get there? This was a very large trial that looked at intensive control of blood pressure with very encouraging findings, suggesting that if you set your target of blood pressure control to 120 rather than 140, you can reduce quite significantly the risks of mild cognitive impairment or dementia, setting a new prerogative to treat blood pressure actively and intensively. Next slide, please. We're building a platform for education for prevention called HALT AD. This is an online platform that's going to provide easy to digest information on the modifiable risk factors that I've shown you. We want to empower, improve literacy, and motivate behavioral change. There will be group support and moderation done monthly by Zoom, and persons will, interacting with this program, be welcome to HALT AD, and they will also be achieved, as they complete questionnaires, will be given a speedometer or a visualization of how their diet looks, how their sleep score looks, areas to concentrate as they navigate through this program that has multiple modules within it. Next slide, please. I want to make you aware that we're going to pilot this online educational platform called HALT AD, which is available in English or Spanish for persons aged 50 to 85 who are well and don't have cognitive impairment or Alzheimer's disease. You need to be able to access the internet, be willing to join the online discussion groups, be able to have sufficient vision and hearing, and be willing to participate for up to 4 months. We see this as a really important opportunity to help us shape this program, which we're very optimistic about. Next slide, please. I mentioned that there's a clinical trial. Dr. Glasgow is serving as the principal investigator at the Adrc for this one. It's in partnership with Dr. Ornish to determine whether early Alzheimer's can be stopped or improved by comprehensive lifestyle program with interventions including diet, you see whole foods, low fat, low sugar, plant-based, moderate exercise, stress management, including meditation, psychosocial support, while we examine cognition, everyday function, biomarkers of vascular health, length of telomeres, which tell us about one's health and aging, and the microbiome. Next slide, please. This one is open and enrolling. So it literally takes a community to do these clinical trials. It takes funding, it takes dedication, and I acknowledge and salute all of our colleagues that contribute to this effort every day. Nadine has just put up in the chat, again, the contact email address, and we hope that this will be of interest and you'll be interesting to join this effort. Thank you.
Wonderful. That is truly inspiring. The full set of talks. We're so delighted that you stuck with us through this difficult period, and thank you, Dr. Glasgow, Dr. Jones, Dr. Zlatar, and Dr. Feldman for such a fantastic summary of some of the incredible work that's still going on here at the ADRC. You can see the pandemic did not stop us, not by a long shot, and but I would say I'm very inspired from that set of presentations, and I hope you, our community of participants, are also inspired. And the most important message of that entire time is thank you to our participants. We couldn't do any of this without you and your brave participation in our programs, and thank you for sticking with us through this difficult period. And we're very much looking forward to our next phase. So I'm here to begin first to give a little bit of a wrap up, and then we're going to open up for questions that were submitted prior to this presentation, and then also some questions that you might have that you can put into the chat for us. So we're delighted, and we are here for you. We are here to give you information, and a couple of the things that we want to highlight: the quality of life programs. Our social work team is available for consultation. Both Yvonne Arias and Tracy Truscott, our valued partners in social work, are available to you. Our support groups that are virtual and in person are available to you. And please remember to register if you're interested in a musical biographies program that you can learn about on the website, the adrc.ucsd.edu, and also the virtual memories at the museum. And please check out our virtual brain blast series, which is on a YouTube channel, the UCSD Shiley-Marcos ADRC, where we provide some updates on new studies that you may be interested in participating with us for those programs. So those are some of our resources. Please come to the website to learn more. Stay connected with us, not only through the website, which has been updated, our monthly e-newsletter, and our that's a new e-blast that we've been doing, and the usual currents newsletter that we do twice a year, and now a Gaceta Edad Spanish annual newsletter, as well as our social media streams on Facebook, Instagram as well. So please follow us, subscribe, etc. So let's go on to our questions at this time. First, I thought we would tackle some of the ones that were submitted in advance, and please go ahead and submit additional questions you might have in the chat. I know that each time we've had these in-person events, it's been very lively and a great discussion happens, and everybody comes up afterwards, and we get to spend time linking and informally answering some of the questions and updates. That's a little more clunky in this format, but the best we can do is to ask you to go ahead and put your questions in the chat, and we'll try to get to each of them. So one that was submitted was, given recent advances in biomarkers and genetic research on Alzheimer's, how should adult children of affected individuals be counseled about screening for themselves? Are there resources at the ADRC that can help guide decision-making and provide advice about the pros and cons of initiating the process? So Doug, would you like to take that one, or I'm happy to take it as well?
Sure. So, these are important questions. And certainly it is possible to get a lot of testing done. Whether it's advisable or helpful or not is a complicated question. In general, we discourage people who are asymptomatic to get tested in any of these settings outside of a research study. There are research studies that are ongoing that are looking at testing, and there are a number of aspects that would be important, namely getting a test done through a reliable laboratory, getting all of the appropriate information to understand your risk before you get tested, having some notion of why you want to be tested, what you'll do based on the results, and then having some process to disclose the result. So it turns out that the National Institute on Aging is very interested in this topic, and there is a very large working group. It goes by the abbreviation of AGREED, that has been working on a number of resources that deal with questions of pre-symptomatic testing for biomarkers and genes. I will dig up one or two position papers and resources that have come out of this so far. There are some comments about when to get tested and in what settings, and in particular about the ApoE gene. There are some good resources about why one might or might not decide to get tested for any of these. And again, right now, I would say we are not recommending any of this outside of a research setting.
Great. Thanks, Doug. And that's a great summary that we've struggled with this topic a lot. There's a lot of interest in getting information. We feel like, genetic counseling may be an important piece, and an interaction with your own personal physician as well, because they may be brought into the process if this does come to fruition in the future. But at this point, not yet within our research setting are we returning such results. So that was one of the questions in the chat. Doug, do you want to take this other one on biomarkers too, on the blood tests?
Okay, so there are a number of new blood tests. There's not just one. I described an amyloid test, and I mentioned several tau tests. The amyloid test that I showed you is available for use in people who have symptoms, who are suspected of having mild cognitive impairment or Alzheimer's disease. And in the data we've seen so far, it is about 80% accurate overall. And that's complicated because the cutoff points have been chosen in a way that a positive result will be accurate, a negative result will be accurate, but a whole bunch of people will have a result that is in between, and that is called indeterminate. And so it's possible to have this test and get back a result where the person who ordered the test will say, well, we really can't tell if you have amyloid in your brain or not. So the other issue with this test is if we test for amyloid alone, it's possible to have amyloid, as I mentioned, 10 to 15 years before symptoms of Alzheimer's disease develop. And so studies have actually done this and looked at normal people in the community, cognitively normal folks in their 80s, for example. It turns out that there's a 20% rate, at least, of having a positive amyloid test, but that doesn't necessarily mean that you have Alzheimer's. It means that you might have a slightly higher lifetime risk of getting Alzheimer's, but that would be an issue. So if you are symptomatic and we order an amyloid test, and you're symptomatic because you have vascular dementia or vascular problems due to strokes or whatever, and someone orders this test and it comes back positive, again, the interpretation has to be done in clinical context in relation to everything else. So any of these tests are just tools. And certainly when we see folks in the memory disorders clinic at UCSD, we try to be as holistic as we can. And we're trying to come up with the most complete and most accurate diagnosis that we can.
Perfect. 100% agree. These are difficult issues to wrestle with. We've put in position papers about the inadequacy of amyloid alone as an approach to say where a person falls on the spectrum, and that's why Doug laid out this ATN process. But even with that, it's as he showed at the early part of the stage of his slides, it's getting even more and more complex as you fold in other etiologies of dementia. So there's so much that needs to be done, and we continue to work on that. And your participation in our studies has been greatly valuable, especially because we're seen as one of the centers as a great resource for validating these markers, because your participation, you're seen as very well characterized individuals, and so we've been a valuable resource for individuals seeking that data to validate these measures. And then also the very robust, because we've been around for almost 40 years, the fact that we have a very robust, well-characterized brain database for individuals that were followed in our center and then generously donated their brains at the end of life is super, super valuable resource, not only for our local community, but for the entire nation, as a lot of people reach out to us. So San Diego for Alzheimer's research is really a hub of scientific advances in these areas. And so it's a very special place to be. So the next question really related to that first portion. So I'll just, we kind of answered about clinical or genetic testing in the research setting. Clinical testing would kind of fall under that same answer. There's a question about the NeuroQuant test, the measurement of brain structure anatomy used in clinical settings, and how does it compare to the PET scan for usefulness in measuring Alzheimer's disease. I was involved in the development of that tool, and so I can tell you that really it's meant to really add a component to the radiological read that relates to neurodegeneration. It's not meant as a standalone diagnostic test. It really is just to highlight areas that might need further attention for the radiologist to look at and say, hey, wait a second, is this person's brain truly at where we would expect it for this person's age? And if not, at least return that information to the clinician so that they can assess it further. It's been built out now also to look at other measurements, like in epilepsy, where the two hippocampi need to be looked at for whether they're symmetric. So there's some very straightforward approaches to using it. It's been stretched probably beyond that. It's been used in a lot of approaches that are outside of its intended use. But in terms of its usefulness in measuring AD, we felt like it was a valuable ancillary tool to help highlight radiologists' attention to a feature that is seen in imaging, which is neurodegeneration, which before this tool was in place, it seemed like a lot of the radiologists were not really paying attention to that aspect of the clinical read. So that's what it was meant for. It's a lot less expensive than doing a PET scan, but a PET scan gives different information. PET scan gives functional information of how the metabolism is active in the brain, and that can be useful for looking at, say, frontotemporal dementia versus Alzheimer's disease. So there's different uses for each of those tools. Okay, let's move to the next. Has any more been learned about the causes of early stage Alzheimer's? Okay, so a lot of us could probably take that on, and I think what you saw not only in Howard's talk, Doug's talk, Dr. Jones' talk, we're all working on the causes of early stage Alzheimer's disease. One would definitely find that the iPSC work is, we hope, going to tremendously propel additional knowledge in these early stage Alzheimer's disease causes. Dr. Feldman, through the Epstein program, just assembled a group that's looking specifically at some of the very genetic forms of Alzheimer's disease, and we are indeed learning a lot. I think that question itself is so full of potential answers that I think the answer is yes. Much more has been learned, but I think it's too much to tackle in this question and answer phase. So Doug, again, sorry to keep going back to Doug, but Doug is running an all-FTD study. So do you want to talk a little bit about the all-FTD study, Doug?
Certainly. So we are part of a national study across the US that is called all-FTD, or all frontotemporal dementia. And we are actively seeking out and enrolling people who have frontotemporal dementia. There are different types of frontotemporal dementia. Some people have behavioral changes, language changes, and others have movement changes. And we're enrolling all sorts of folks with those kinds of changes. If they have a diagnosis of frontotemporal dementia, they undergo brain imaging, cognitive testing, neurological testing, blood draws, DNA testing, and in some folks, we're doing lumbar punctures and then following people over time. And again, lots of different aspects of this research. These include looking at genetics of frontotemporal dementia, looking at biomarkers, trying to see how to characterize people best over time. And there are some clinical trials that are starting to emerge for frontotemporal dementia as well.
Great. Thank you. So the next question is about the brain scans that were stopped due to funding issues. We used to run, for a period of time, we were scanning every new individual as soon as they enrolled. It turned out that we had so many brain imaging researchers that were seeking to image our individuals on their own through the collaboration with their research, that we felt like we were gaining a lot of brain scans even without doing it in that initial visit. So that continues to be our process, but there is an exciting new funding that did come in that does allow us to pursue brain scans in our select individuals in our cohort, called the SCAN Initiative, and also the DVCID, which was discussed briefly, the diverse vascular cognitive impairment and dementia. So there are some new funding opportunities that we have been fortunate to have been awarded that is going to increase our brain scanning pursuit. So that's good news that's coming up. Howard, would you like to take this next one? Which clinical trial is showing the most promise for people with ApoE4 and limited ARIA occurrence?
So it's a great question. So to clarify, at the moment, ARIA is a phenomenon that is in longhand amyloid-related neuroimaging abnormalities, and it occurs when we use amyloid-lowering treatments. So at the moment, when we have therapeutics, as I showed earlier in the slide, that hit, for example, a tau target, or hit a mitochondrial target, or hit a metabolic target, we have no expectation that that will cause ARIA. So we know there's an association of ApoE4 as a risk factor for ARIA in persons with amyloid-lowering treatment. But in terms of promise, there are many therapeutic avenues for which we would not foresee a risk of ARIA, and those would be perhaps the way to answer this question.
Thank you so much. So the next one is, what have been the progress of and findings of the UCSD Alzheimer's study? I presume you mean we have many Alzheimer's studies, but the ADRC longitudinal study, you got a highlight in this presentation. I wanted to bring up one additional associated program, that is the microbiome project, the AGMP project, that a lot of you have participated in, where a stool sample is sent in for looking at microbiome association with Alzheimer's disease. That's been extraordinarily helpful. We are, as at UCSD, we have Dr. Rob Knight, who is one of the world leaders in microbiome studies. So we're very proud to be a main enroller for that study across the nation. And so we're certainly going to be reaching out for year 2 samples, and also enrolling diagnosed individuals, people who do have some impairment in cognition, for year 1 studies. So those of you that participated, thank you so much. And we will likely be reaching out for another sample for a second year. And for any individual that that has some diagnosis of cognitive impairment, we may be reaching out to you for year 1 as well. So thank you for all that. The next question is about the Tau Tangles and Cannabinoids. This is actually, a few of us know about this finding, and it was presented to the ADCS, Dr. Schubert's finding of amyloid and maybe some tau removed in mice studies. I did try to do a quick update on that, to look through the literature, seeing this study, to see whether any human studies have been launched. I have not seen them. If anybody else know, Howard, go ahead.
So Canadian colleagues have run a trial with nabilone, which is active on the cannabinoid receptors. I don't know if it was specific to a tau outcome, but they tested it in agitation, so in more advanced Alzheimer's. And colleagues Nathan Herman reported that it was beneficial in a small, controlled, randomized clinical trial. So we're beginning to get little bits of information. It has the potential for negative cognitive effects, but it had the benefit on agitation in this one study.
Thank you so much for that. Yes, and the other pieces that were pathologically tied were the psychoactive components that were seen in these cannabinoids. So there's a whole host of continuing research in that. I have not seen, at least out of the Salk study, the human translation at this point. One question about breakthroughs in Alzheimer's disease in Israel. My connection with specifically Israel studies comes through the Weizmann Institute, where I did Fulbright for a year before starting this endeavor. And so I'm mainly familiar with the Weizmann Institute's breakthroughs. And one place where they're particularly strong is the interaction between the immune system and the brain. They've always had a really innovative approach to looking at immune interactions with not only in Alzheimer's disease, but other illnesses. And Michal Schwartz from the Weizmann Institute and Yoni Kipnis, who came to the United States from Michal Schwartz's lab, are really leading a lot of studies in this newly accepted idea that the immune response of the brain is very important in the development of Alzheimer's disease and in the process of Alzheimer's disease. So let's move to the next slide. Okay, water fasting. I was not able to find a lot on water fasting. I think in general, staying away from water is usually seen as a bad thing in any aged individual. We like to keep people hydrated. That's the standard approach for caring for individuals. We mostly have problems with people being dehydrated as they age. And so I would not go after a water fasting approach, although I don't know the details of this. If anybody else has anything else, they can jump in. But otherwise, we'll move to the next one, which is quite... Oh, go ahead. Yeah.
So Howard had a comment maybe about time limited feeding, maybe.
That's a very exciting thing.
Yeah, so that's what I was
going. Watts has some very interesting pieces here, and Howard, go ahead and talk about.
Well, I was just going to say that feeding. I want to reinforce that for older persons, we don't recommend water fasting. It's not good for the kidneys, and really, there's no evidence currently. I just would not want people to think it's a viable approach. On the other hand, there's been some really interesting research on generating mild ketogenic states by shifting one's eating patterns. And Dr. Pam Taub at the Cardiology Center has looked at shifting so that people don't eat after 6 in the evening and have a 12-hour fast to the morning. And those results have looked quite interesting, and ketones are potentially a brain food. Having said that, not for persons that have symptoms of Alzheimer's disease, but perhaps for those looking at modifying risk factors on the way. So just so that we don't get into the wrong space here.
Exactly. Excellent. And also Dr. Paula Desplats from the Neurosciences Department has been looking in mouse models of the intermittent fasting as well, which it seems, again, stay tuned. These are very interesting potential non-pharmacological approaches, not the water fasting, but potentially the intermittent food fasting, but again, has not been studied systematically in the way that it needs to before making a recommendation. Because a lot of times these approaches look promising and they may have adverse effects. So don't do it without the data. The next part is an important aspect that we tried, and the UCSD Adrc did put out a publication that talked about these anticholinergic medications, including the doxylamine, succinate, and then in the chat here talking about Benadryl. Those are both anticholinergic or diphenhydramine, which is Benadryl, not causing Alzheimer's disease, but certainly exacerbating the deprivation of a very important neurotransmitter in the brain, acetylcholine. So most of the medications we use in the symptomatic management of Alzheimer's disease are trying to boost up acetylcholine in the brain. And both of these medications that are mentioned, the diphenhydramine and the doxylamine succinate, are medications that do the opposite. So indeed would be expected to contribute, at least in the short term, while on the medication, decrease short-term memory. It does, and it gets complex to analyze it, to say, well, how much of that is then going to lead to later cognitive decline? Is it mostly just a challenge to the brain for those that are already showing some mild impacts of aging and just need every bit of acetylcholine that they can get a hold of? And so in most of clinical practice, we're very careful in the aged population of using such anticholinergic medications, including bladder medications that may be anticholinergic. So does it have the same effect in a 50-year-old person as it does in an 80-year-old person? And the answer is less of an effect in a 50-year-old person, at least in the acute observable symptoms that we see when a younger person takes these medications. They tend to tolerate it quite well. An elderly person, we tend to avoid them in medicine. If anybody else wants to jump in and add anything to that, but I think I covered it okay. Well, I think the opinions on the Biogen drug have been covered by Howard's very excellent slides. And rosemary extract, also untested approach that certainly if, again, that's what we do. There are many, many potential opportunities that need to be studied, and we don't encourage using these things without the research that needs to be done in humans to actually explore it. So that's kind of what I have on any, not just rosemary extract, but many of the different vitamin approaches or other natural therapies that have been proposed. Any additional slides here?
So Dr. Feldman has a comment about
rosemary. Go ahead,
extract.
Howard.
Probably a comment about the Epstein.
Yeah, thank you. So just the audience might be interested to know that as part of the Epstein collaboration, we are taking an interest in these natural products, and more specifically, the underlying pharmacology, the evidence that there is to support their use. And if they look promising, we're hoping, and if we can produce them in pharmaceutical grade material, we're hoping to launch small, smart clinical trials with them. So stay tuned, we're on it, we're aware, and it's a very interesting opportunity to pursue.
Super, and we are at 11:30, and I just want to end and wrap this up by really, really giving from the bottom of our hearts, a thank you to all of you who spent your time with us through these challenging periods. Stay with us, stay tuned, stay engaged with us. We thank you for your connection to the Shiley-Marcos Alzheimer's Disease Research Center. I hope you enjoyed our presentation. Next year, we will. This is usually the part where we all gather at the front and just have great conversations. And I'm sorry that's not going to happen this year, but hopefully the next time we'll be able to do that. So thank you all for your attention.